I'm Romila Lemos, Assistant Professor, Department of Forensic Biology, Institute of Forensic Science, Mumbai. Today's topic of our discussion and learning is all about forensic serology. This is the part 2. We will be covering the following modules in today's lecture. Module 1, reaction between antigen and antibody, that is the primary reaction. Module 2, reaction between antigen and antibody, which is the secondary reaction. Module 3, sample collection of serological evidences from scene of crime. Module 4, sample collection of serological evidences from suspect and victim. Module 5, biological health hazards. Module 6, precautions. And finally, Module 7, conclusion. Let us begin with Module 1, reaction between antigen and antibody, that is the primary reaction. Now let us discuss the antigen and antibody binding reactions. The binding of an antigen to an antibody is an equilibrium reaction consisting of three types of reactions: primary, secondary and tertiary. The primary and secondary reactions form the basis for many forensic serological assays and will be discussed in the following subsections. The third type is called the tertiary reaction. It is used to measure in vivo immune responses such as inflammation and phagocytosis. Because most forensic serology tests are in vitro assays, the tertiary reaction is not commonly utilized in forensic serology testing and will not be discussed here. Let us take a look at the primary reactions. A primary reaction is the initial binding of a single epitope of an antigen and a single binding site of an antibody to form an antigen-antibody complex. This is a rapid and a reversible binding reaction and can be expressed as shown. At equilibrium, the strength of the interaction can be expressed as the affinity constant, that is the Ka, that reflects the affinity of binding, where the square brackets indicate the concentration of each component at equilibrium. Ka is the reciprocal of the concentration of free isotopes when half the antigen binding sites are occupied. Thus, a higher Ka corresponds to a stronger binding interaction. Techniques such as enzyme immunoassays, immune fluorescence assays, radioimmunoassays and dye-labeled immune chromatography can measure the concentrations of antigen-antibody complexes formed by such kind of primary reactions. These techniques are the most sensitive for detecting the presence of an antigen and an antibody in a sample. Module 2, reaction between antigen and antibody, that is the secondary reaction. The primary reaction between an antigen and an antibody is often followed by a secondary reaction. The three types of secondary reactions are precipitation, agglutination and complement fixation. The techniques that detect secondary reactions are usually less sensitive but easier to perform than primary reaction assays. The precipitation and agglutination reactions form the basis for many serological assays performed in forensic laboratories. These reactions will be discussed in the following subsections in detail. The third type of reaction is called complement fixation. If an antigen is located on a cell surface, the binding of the antigen and the antibody may activate the classical complement pathway and lead to cell lysis, also known as a complement fixation reaction. The detection of this type of reaction is not commonly used in forensic serology. So let us go on to precipitation. If a soluble antigen is mixed and incubated with its antibody, the antigen-antibody complexes can form cross-linked complexes at the optimal ratio of antigen to antibody concentration. The cross-linked complex is insoluble and eventually forms a precipitate that settles to the bottom of a test tube. Antibodies that produce such precipitation are also called as precipitins. Agglutination. As discussed in the previous subsection, precipitation reactions involve soluble antigens. If the antigens are located on the surfaces of cell or carriers such as carrier cells such as the sheep erythrocytes, bacteria or latex particles, then such antibodies can bind to the surface antigens and can form cross links among cells or carriers, causing them to aggregate. This aggregation is referred to as agglutination. In agglutination, a visible clumping can be observed as an indicator of the reaction of the antigen and the antibody. If the antigen is located on an erythrocyte, the agglutination reaction is designated as heme agglutination. Agglutination is a two-step process that includes initial binding and lattice formation. In Module 3, we will be discussing the sample collection of serological evidences from the scene of crime. Evidence may be recovered from many sites: the crime scene, vehicle involved in the crime, the suspect's body and clothing, and the victim's body and clothing. In sexual assault cases, evidence such as penile swabs from a suspect, the suspect's underwear for victim's DNA, and fingernail or tip samples may be useful evidence. When appropriate, collect as much evidence as possible as quickly as possible from the bodies of the victim and suspect. Biological evidence is fragile and can easily be destroyed. The recognition and recovery of such evidence must be performed properly by the investigator in order to make the best use of it. Let us take a look at the different kinds of specimen or samples available at the crime scenes. First one, liquid blood specimens at the crime scene. Liquid blood should be collected with a clean, preferably sterile syringe or disposable pipette and transferred to a clean, that is sterile, test tube. A blood clot can be transferred to a clean test tube with a clean spatula. A clean cotton cloth can be used to soak up liquid blood or a blood clot, avoiding areas containing only serum. Wet blood samples, if they are collected, must be preserved in a suitable anticoagulant and kept in a refrigerator. These specimens should be submitted to the laboratory as soon as possible. It is very important to label the specimens with case number, item number, date, time, location, and the evidence collector's name. The second kind of evidence that may be found at the crime scene is wet blood stains. Small objects bearing wet blood stains should be allowed to air dry and then collected as it is. An effort should be made to preserve the integrity of any blood stain patterns during packaging and transportation. Large objects that cannot be removed from the crime scene may have wet blood stains on them. The wet blood stains should be transferred onto clean cotton cloth. Blood-stained cotton cloth must then be allowed to air dry before packaging in a proper container. Each object and container must be properly labeled. The next kind of biological evidence that may be found are the dried blood stains on removable items. Dried blood stains on weapons, garments, and other movable objects should be collected separately by collecting the entire item. Each item should then be placed in its own paper container, and then these should be sealed and labeled properly. Next is dried blood stains on solid, non-absorbent surfaces of immovable objects. The blood stain pattern should be documented and sketched to the extent necessary. The stain can be tape lifted or scraped off the object onto a clean piece of paper. The tape lifter or the paper with blood crust can then be placed into a druggist fold and placed in an envelope which is sealed. Each item must be labeled properly. If the blood stain cannot be scraped off or the support object cannot be cut, then the blood stain may be eluted onto a clean switch, moistened with sterilized saline or water by rubbing the cotton switch on the stained area. The switch is then allowed to dry and is placed in a paper fold packet. The packet is then placed in an envelope which is sealed and properly labeled. It is essential to always obtain a control by repeating the procedure on an adjacent but unstained area of the surface containing the blood stain. If the blood stain is located on an object that can be cut, then a portion of the item containing the blood stain can be removed by cutting with a clean, sharp instrument. Each cutting should be packaged separately and labeled accordingly. An unstained portion of the item should be collected and packaged as a control. The next kind of biological evidence that may be found are semen and seminal stains. It is important to document the semen evidence by notes, photography, videotape and sketching. It is essential to use a clean syringe or disposable pipette to transfer liquid semen to a clean, sterile test tube. These tubes must be labeled with the case and item number, date, time, location and the name of the collector. The specimen should be refrigerated and submitted to the laboratory as soon as possible. Alternatively, liquid semen can be transferred onto a clean cotton cloth by absorption. The cloth is then air dried, packaged, sealed and labeled properly. Seminal stains on panties, clothing, bed sheets, pillows and other movable objects should be collected as it is. If an article has a wet stain on it, the stain must be allowed to first air dry thoroughly prior to the collection of the article. If the stain is on a large object that can be cut, then the stained area should be cut using a scalpel or scissors. If the stain is on immovable non-absorbent surfaces, then the stain should be scraped using a clean scalpel onto clean paper and the paper should be folded into a druggist fold container. Each item should be packaged separately in a clean paper container. Each item's packaging must be properly sealed and labeled. Packaged items should be refrigerated if possible and submitted to the laboratory as soon as possible. Next, we look at the collection of urine, saliva and other body fluids. The collection of these body fluids is done in the same manner as blood and blood stains. Let us move on to the module 4, which is sample collection of serological evidences from suspect and victim. How do we collect blood from a person? It is always necessary to collect reference samples from suspects and victims. In the majority of cases, these samples consist of liquid blood. Liquid blood from a person should be collected by qualified medical personnel. The crime laboratory should be informed if the subject had recently received a blood transfusion of any kind. 2 tubes of blood, about 5 milliliters each, should be collected in vacutainers with EDTA as anticoagulant. In the case of collecting reference samples from post-mortem subjects, a blood sample should be obtained from non-body cavity areas such as heart or major internal blood vessels. Each tube should be labeled with the date, time, subject's name, location, collector's name, case number and exhibit number. Blood samples must be refrigerated, not frozen, and submitted to the laboratory as soon as possible. To collect seminal evidence from sexual assault victims, when dealing with sex crimes, the victim should be taken to the hospital immediately and the examination started as soon as possible. Photographs should be taken to document any injuries which the victim received. If necessary, oral, vaginal and/or anal swabs should be taken from the victim and air dried for 1 hour in a moving air source as soon as possible. They should be collected as soon as possible because the body begins breaking down the various components in seminal fluid through drainage, enzyme activity, pH variance, etc. The swabs should be air dried under a fan for at least 1 hour. This can either be accomplished by the doctor at the hospital or upon collecting the kit from the doctor, the investigator should bring it immediately to a secure place and air dry it. The reason for this is that the moisture in the swabs allows microorganisms to grow which can destroy the evidentiary value of the swabs. To collect tissue, organ or bone samples. Each item of evidence should be described in notes and documented by photography, sketches or videotaping. This type of evidence item can be picked up with a clean pair of forceps. Each item should be placed in a clean container without any added fixatives. Each container should then be properly sealed and labeled and stored in a freezer. Evidence should be submitted to the laboratory as soon as possible. When collecting reference samples from post-mortem subjects, if the body has decomposed, in addition to the blood sample, it is mandatory to collect as many of the following specimens as possible, such as a portion of deep muscle tissue, certain organ tissue, example heart or brain, 2 to 4 intact molar teeth. If identification is an issue, ensure that the mouth X-rays have been taken, and a sample of compact bone such as femur. The specimens collected should be away from the site of injury. That is, if suppose a head injury is seen, the sample of the brain tissue must not be taken. It is also essential to immediately freeze these samples without addition of any preservatives such as formalin. In the module 5, we will now be discussing the biological health hazards. Some of the chemicals used for preliminary assays at the crime scene are carcinogenic, that is, they may cause cancer, while others may cause long-range health hazards. Biological fluids such as blood, semen, saliva and urine are handled on a routine basis. These fluids can contain any of the following diseases such as AIDS, Hepatitis B, measles, gonorrhea, syphilis and tuberculosis. This becomes a risk not only of the investigator but also that of the laboratory personnel who are handling these fluids and the investigator's own family. In the module 6, we shall discuss the precautions that are necessary to handle the biological fluids. Universal precautions. It is not possible to determine if every bodily fluid or stain collected from crime scenes is contaminated with a blood-borne pathogen. Therefore, all bodily fluids and tissues are presumed to be contaminated. When individuals handle any type of biological evidence, procedures need to be in place to reduce or eliminate the risk of exposure to blood-borne pathogens that can transmit disease. Common diseases or viruses caused by exposure to blood-borne pathogens include hepatitis and HIV. These raise the most concern because of the potential for lifelong infection and the risk of death associated with infection once an individual is exposed. Personal protective equipment. The appropriate use of PPE or the personal protective equipment is intended to protect the individual and the evidence from cross-contamination. PPE includes disposable gloves, disposable overalls, laboratory coats, masks, and eye protection. PPE should be used in every situation in which there is a possibility of exposure to blood or infectious diseases. Gloves and protective clothing should be worn when providing first aid or medical care, handling soiled materials or equipment, and cleaning up spills of hazardous materials. Face protectors such as splash goggles should be worn to protect against items that may splash, splatter, or spray. PPE must be clean and in good repair. PPE that is torn or punctured or that has lost its ability to function as an effective barrier should not be used. Disposable PPE should not be reused under any circumstances. While using PPE, individuals should not touch their eyes or nose with gloves. PPE must be removed when it becomes contaminated and before leaving the work area. Used protective clothing and equipment must be placed in designated areas for storage, decontamination, and disposal. Dried blood or other potentially infectious material should not be assumed to be safe. PPE should be used when handling these items. When wet material is spilled, the area containing blood or other potentially infectious material should be covered with paper towels or rags doused with a disinfectant solution such as 10% bleach solution, left for at least 10 minutes and then removed. Materials should then be placed in a waste disposal bag designated for biohazardous material. Appropriate PPE should be used throughout this process. Hazardous biological evidence packages must be appropriately labeled with biohazard labels and signage. Without the biohazard label, other employees could inadvertently be exposed to risk or could contaminate the evidence. The labeling and signage guidance also applies to any shelves or rooms where these items are being stored. Additionally, a ventilation system may be required to ensure that employees are working in a safe workplace. Exposure control plan. Crime laboratories, property and evidence rooms and other locations where biological evidence is stored should have exposure control plans in place that are designed to minimize or eliminate occupational exposure to blood-borne pathogens. An exposure control plan is an employer's written policy outlines the protective measures the employer takes to eliminate or minimize employee exposure to blood and potentially infectious diseases. Biological evidence disposal regulated waste. Regulated waste is liquid or semi-liquid blood or other potentially infectious materials, contaminated items that would release blood or other potentially infectious materials in a liquid or semi-liquid state. If compressed, items that are caked with dried blood or other potentially infectious materials and are capable of releasing these materials during handling, contaminated sharps and pathological and microbiological wastes containing blood or other potentially infectious materials. It is required that regulated waste be rendered non-infectious prior to its disposal. Once the biohazard is decontaminated, it can be disposed of like any other solid waste. Staging for destruction or decontamination. Items to be destroyed or decontaminated must be removed from the active inventory and staged in an area for bio-items that are scheduled for destruction and appropriate disposal. There are several methods to be used to destroy or decontaminate biohazardous material. First one is incineration. Incineration involves the actual burning of the waste. This method both destroys and decontaminates the evidence. Although effective, incineration is associated with serious air quality concerns. Evidence handlers should consult local and state laws for guidance. The second one is the thermal treatment. Similar to incineration, thermal treatments use heat to destroy any pathogens present in the biological material. There are several types of thermal treatments such as autoclaves, microwaves and dry heat systems. Each of these can be used to render biological evidence safe prior to its disposal. The third one is the chemical treatment. The most common method of decontamination, use of chlorine either in the form of sodium hypochlorite solution commonly known as bleach or in the form of the more powerful and correspondingly more hazardous gas chlorine dioxide. These compounds are relatively cheap and effective. Individuals responsible for destroying or decontaminating evidence should consult the state regulations and the crime laboratory before deciding on an appropriate and safe method for destroying or decontaminating the evidence. Module 7 Conclusion. Thus, a major part of the crime scene investigation relates to the understanding of the biological evidences that are to be collected and their proper sampling and packaging while taking all the necessary safety precautions. With all these information, here we come to the end of today's lecture. Do keep in mind what we discussed today. It is time for you all to do some self-study. This is Romila Lemos signing off. If you want to learn more and enhance your knowledge, you may log on to our website for MCQ, quizzes, LORs, that is www.cec.nic.in. Till then, goodbye.